Pemphigus vulgaris is a potentially fatal autoimmune skin disease caused by a person's own antibodies attacking cell adhesion structures called desmosomes. Corticosteroids and other drugs have increased survival dramatically, but risk of fatal infections and adverse effects related to these treatments not only impact the quality of life for Pemphigus patients, but all individuals with autoimmune diseases who rely on these treatments. Recently, a new method of treatment utilizing chimeric autoantibody receptor T cells, or CAR-Ts, was shown to be safe and effective in mouse models. Let's first focus on what goes wrong in Pemphigus before moving on to learn about how CAR-Ts work. Pemphigus as a term refers to a group of relatively rare skin diseases including Pemphigus foliaceus, paraneoplastic, IgA, and what we will focus on, vulgaris. Basically, Pemphigus vulgaris is excessive and intense blistering of your lower epidermis and mucosal membranes. This is noticed under a microscope or histologically by a phenomenon called acantholysis, which is just the loss of inner cell adhesion, which leads to blistering. Normally, your skin cells are attached to each other through several means, one of which involves complexes called desmosomes. Desmosomes form the linkage between cells and anchor cells to cytoskeletal networks. They are comprised of three classes of proteins: desmoglians and desmocollins, which fall under the more general class of calcium-dependent adhesion proteins called cadherins; placoglobin and placophilin, which fall under the more general class of armadillo proteins; and placins. Desmoglian and desmocollin, the cadherins involved in Pemphigus vulgaris, consist of 5 domains, which each contain clusters of beta strands that form a pseudo-Greek key motif. Domains are held together and rigidified by calcium ions that bind to charged amino acids at the end of each domain. 2 cadherins interact through a strand swap interface, where the tryptophan residue of one cadherin inserts into the hydrophobic pocket of a different cadherin. In Pemphigus vulgaris, your own antibodies become reactive to these adhesion proteins, specifically desmoglian. They bind primarily to the amino end of desmoglians, which are directly involved in the strand swapping phenomenon that holds keratinocytes in your skin together. Autoantibodies block the tryptophan interactions, which are involved in the strand swapping mechanism. A lot of the biochemical characterization of Pemphigus vulgaris was done in the 1980s and 90s by Drs. Stanley and Emagai. Their initial experiments have been confirmed by crystallographic studies, electron tomography, electron microscopy, and atomic force microscopy. Current research at the University of Pennsylvania has led to a method of treatment for Pemphigus vulgaris inspired by the recent CAR T-cell cancer therapy, also fostered at Penn. Ellebrecht and others created a T-cell harboring a lab-made receptor which is attacked by problematic B cells. This receptor mimics the protein domain of desmoglian that is targeted in Pemphigus vulgaris. After being attacked by the autoreactive B cells at their new receptors, these re-engineered T cells are then able to destroy the B cells that cause Pemphigus. Ellebrecht and others also demonstrated that these T cells show virtually no toxic effects and led to long-term remission of the disease. This could mark a transformation in how Pemphigus and other debilitating autoimmune ailments are treated. Pemphigus vulgaris is definitely not a household name, but thanks to the initial work of Stanley, Emagai, and others, many new scientists have been inspired to learn more about what's going on in Pemphigus. Several hurdles still remain in the way of the CAR-T therapy that was developed by Ellebrecht, but the biggest one being human clinical trials. My own work in the lab focuses on characterizing a canine model of Pemphigus that will hopefully solidify CAR-T's efficacy and speed its progress through the human clinical trials. Through the creation of domain-swapped desmoglian and desmocollin protein constructs, we are going to pinpoint which portion of the cadherins in canines are targeted in Pemphigus. This will allow us to create and test a canine CAR T cell that will hopefully help dogs suffering from Pemphigus, but also legitimize that this treatment actually works and will probably still work in humans as well. With further work, the promising initial studies of the CAR T therapy can lead to a revolution in the quality of life for those who suffer from Pemphigus and all autoimmune diseases.